So the next thing that we're going to discuss is going to be the applications of DNR, or of PCR, so how we analyze those DNA segments. So the application of PCR can be used for a lot of different things. It can be used to identify individuals by using short tandem repeats. Short tandem repeats is a way that we can sort of fingerprint or analyze DNA traces. So DNA is going to be treated with restriction enzymes that are going to dice and chop up DNA into unique fragments. Since our regions that correspond with the DNA restriction enzymes are unique to each individual, the fragments produced are also going to be unique to each individual. A process known as DNA electrophoresis can separate the fragments according to size and charge, which will produce a unique pattern known as a DNA fingerprint. It's usually used to measure the number of repeats of short sequences. It can be used in paternity suits, rape cases, corpus ID, so on and so forth. Here is what a gel electrophoresis can look like. We can take the mother's DNA and we can chop it up using or chop up the short tandem repeats and then run them through a gel to see how they separate out. Now what we can see here is we have the mother's DNA, we have the child's DNA, and then we can try and see we have two males who may possibly be the father. Now because the child gets half of its DNA from its mother and half of its DNA from its father, a child's banding pattern should line up with either the mother or father at any given band or at any given point. We should see each one of these bands in the child should appear on either the mother or the father. So this first band that appears on the child matches both the mother's band and both father's bands. So this is some sort of genetic information that is common to all humans. The next pattern, or the next band that the child has, matches with a band that is found over here on male 1. This band also matches a band that's found on male 1. This band here matches the mother's DNA. This band matches the mother's DNA. This band matches, it looks like not a mother. It does match male 1, does not really match male 2. Male 2 is a little bit off center from this band. This band here does match male 1. This band here matches the mother, does not match any of the males. This band matches the mother, none of the males. This band matches male 1, maybe male 2, but it really does look a little bit off center, so it's hard to tell. This band matches male 1, and that is all for the child. So based on that information, based on this gel electrophoresis that you're seeing right here, would you say that this child belongs to male 1 or male 2? And I'm going to leave that out there for you to decide. So that's going to be the process of gel electrophoresis. Now, gel electrophoresis isn't really used anymore. Instead, what we use are fluorescent labels that are used to tag the regions of DNA, and then an automatic readout of fluorescence is going to be documented. So now, automated DNA fingerprinting looks a little bit more like this, but we're still going to see specific fluorescent patterns that are going to relate or correspond to either the mother or the father, if we are in the case of paternity. In any case, that DNA profile is going to be completely unique to that particular individual.